Hello, I'm Jhon D Ibloguen, and we are going to talk about the antibacterial agents. Bacterial infections and diseases are caused by the pathogenic bacteria. The main role of antibacterial agents is to cure the bacterial infections and diseases by killing or stopping the bacteria from growing. One way to fight against diseases causing bacteria, especially when they enter the body, is through the administration of antibiotics. Looking back on the history of the antibiotics, when Sir Alexander Fleming accidentally discovered the antibacterial agent penicillin from Penicillium notatum in 1928, that resulted to a revolutionary discovery leading to the rise of the field of the microbiology and its uses. In veterinary medicine, the use of antibacterial agents are also common in treating diseases caused by bacteria in infected animals. There are 5 classifications of antibacterials. Number 1, type of action. Number 2, source. Number 3, spectrum of activity. Number 4, chemical structure. And number 5, function. Number 1, type of action. Bacteriostatic and bactericidal. These are 2 types of antibacterials. Bacteriostatics. Bacteriostatics limit the growth or reproduction of the bacteria by interfering with bacterial protein production, DNA replication, or other aspects of bacterial cellular metabolism. On the other hand, bactericidals kill the bacteria outright. This can be done by targeting the bacterial cell wall synthesis or by inhibiting bacterial enzymes or protein translation. To easily remember the type of action, bacteriostatic antibacterial prevents the growth of bacteria, and bactericidal antibacterial kills the bacteria. Examples: bacteriostatic antibactericidals: sulfonamides, erythromycin, linezolid, and tetracycline, etc. These agents inhibit the synthesis of bacteria. Bactericidal antibacterials: penicillins, carbapenems, gentamicin, and quinolones, etc. Number 2, classification based on source of antibacterial agents or simply the source. Antibacterials can be derived from natural sources, synthetic and semi-synthetic sources. Examples: natural sources: cephalosporins, cefamycins, benzylpenicillin, and gentamicin. Natural antibiotics or antibacterials often exhibit high toxicity than synthetic ones. For the synthetic, we have the moxifloxacin and norfloxacin. They are designed to have greater effectiveness and less toxicity. For the semi-synthetic, we have ampicillin and amikacin. Number 3, spectrum of activity. Antibacterials can also be classified as narrow or broad. The identification of narrow spectrum and broad spectrum antibacterials are based on its use in antibiotic history. Narrow spectrum antibacterials can work on a narrow range of microorganisms, meaning if we have a gram-negative and a gram-positive bacteria, narrow spectrum antibacterials can work only on one type, but if we say broad spectrum antibacterial, for example carbapenems, that is a broad spectrum antibacterial, it can work against both positive and negative bacteria. However, narrow spectrum antibacterials are more preferred than broad spectrum antibacterials. Why? Because narrow spectrum antibacterials do not kill as many of normal microorganisms in the body as the broad spectrum antibiotics, meaning it has less ability to cause superinfection, and because it only deals with a specific bacteria, it can cause less resistance to the bacteria. Number 4, chemical structure. Classification of antibacterials based on their chemical structure is very important because it can predict the pattern of toxicity, effectiveness, and other related properties. Antibacterials is divided into 2 groups: Group A, or the beta-lactams, and Group B for the aminoglycosides, which are further classified in groups macrolides, quinolones, and fluoroquinolones, streptogrammin antibiotics, sulfonamides, tetracyclines, and nitromidazoles. Number 5, function. For antibacterials, functions can be divided into 4 groups. Number 1, cell wall synthesis inhibitors. Number 2, inhibitors of membrane function. Number 3, inhibitors of protein synthesis. Number 4, inhibitors of nucleic acid. To simply understand this, we have number 1, cell wall synthesis inhibitors is the peptidoglycan, because these inhibitors works on the cell wall, and cell walls have a polysaccharide backbone called peptidoglycan. Number 2, inhibitors of membrane function is cytoplasmic membrane. When there is colonization of bacteria in the cell, these inhibitors can prevent the cytoplasmic membrane from self-destruction. Number 3, inhibitors of protein synthesis, which is very important for a functioning cell. Protein synthesis is an important target for drugs as it helps in the cure of infectious disease caused by pathogenic bacteria. Number 4. Inhibitors of nucleic acid is the nucleic acid synthesis, which is also an important target of antibiotics. Inhibitors help in inhibiting the growth of new bacteria by interfering with its synthesis. Thank you so much and stand by for the next reporter.